Hello, g'day, good evening, thanks for listening. This is an agricultural and horticultural show focusing on plant science, soil science and microbiology. The information is for educational and scientific purposes, so it's fact checked, and any papers or websites and stuff that's been discussed will be put in the description below. And I think today is going to be part 2 on the What a Plant Knows course. So I'll say g'day to GMF. He says, good evening, peeps. I hope you are well. Aussie, I hope you're healing well, sir. Oh, thank you, mate. Yes, aloe vera does magical things. Doing pretty good. I had an interesting, just before we start, give it 60 seconds odd for other folk to get here. I had a pretty cool discussion with my microbiology, biotechnology professor last night. And it's about herbicides, pesticides and stuff. Glyphosate or Roundup in particular, and the effects on humans, animals, and microbes. And in Australia, they use it rampant. I'm going to probably do a full show on it because he's gone and sent me a lot of papers that say exactly what the truth is, but yet when I search it here from Australia through the search engines, I get nothing or virtually nothing. It all says conflicting, and this is conflicting, but it's not. There's been studies that have been done, and I've got the papers now, because my professor, he sent me heaps of them, and also their research students, they're doing doctorates, sent me a fair few too. So I've only been through 2, and already 2 of them, it's just perfect as exactly what it is, how it affects us. It's actually in your, it was a bit worse than what I thought too, GMF, that in the USA, it's found in some wheats and oats and foods and stuff at a particularly low rate, nano and picogram, but it's still there. And this is a carcinogenic substance. So it's, I think it's worthwhile doing a whole show on, because I've got enough papers to go through, and we can just go through the papers online. Just read them with me. If you're interested in saving your microbes, preserving them, because yeah, the half-life from what people think in Australia is very short. But no, from scientific studies, it proves that it can be up to a year long, and depends on what substrate it is. It could even be up to a couple of years, how things cycle. Hello, 420 Fred. So yeah, I'm just amazed to just get the truth. I just like the science stuff, and just how bad it is for it works on a shikimate pathway. Actually, I'm not even going to go into it here. It's in the metabolic process, and that's for humans and animals. It's just a shame. But Aussie Jeffro probably knows about it. Glyphosate. Hello, everyone, says Aussie Jeffro. Cheers, Aussie CC. G'day, mate. You heard of glyphosate, Aussie Jeffro? It's a shame you didn't. Australia uses it bad. The people that run the country, they love it. They just spray it everywhere. Even the commercial and wholesale farmers love it. And all the search engines say there's nothing much on it I could find in Australia. But from talking to my professor in India, he sent me all these papers. I got at least probably 20 papers to go through now that say all the cool stuff. Hey, there's someone over here from Wild Green Bicycles. Says, cheers, hope all is well. Hope you're doing well too, Mr. Patrick. You might like today's show. It's part 2 of What a Plant Knows, on how it smells, on how it hears, on how it feels touch, how it has a memory, those things. It's pretty cool. It was a course that I did a few years ago, and university course, and I'm just going through all the slides in that. It's pretty cool. So that's what today's on. So I think I've talked enough about herbicides and microbes, but I'm going to make a show for that in a few weeks' time. and go through all the papers, because it affects us humans as well, and animals. And if the Australian government isn't even allowing people to find this information in Australia for some weird reason, it's got to be known. It's carcinogenic, and they spray it, and it's got a half-life, stays around for ages. So it goes into the systems, feeds thing on, animals walk across it, transmit it, things eat it. It's just women sad. Glyphosate, pesticides, don't use them. Use biopesticides. They work so much better. Anyway, when that's not the topic today, the topic is what a plant knows. See how much I could do. What's that? 6 minutes already just talking on the microbes. I love that topic. 2 of my courses this semester are on microbes. The other one's on crop breeding and plant biotechnology. In my 3 courses this semester. All right, let's get into it. Actually, this is pretty interesting, that how long it takes for a carrot seed to form. So I've just gone and processed quite a few thousand carrot seeds. So yeah, 63 days. There you go. But it comes out a little bit different from that. Anyway, we're not talking about that. We're talking about all these slides. See if I've already. There they are. So we're up to halfway. I remember these fibres. So we got this second half to go, right down to the end. And this is what was on last week's, all that stuff up the top. All right, let's go. What am I doing? This one. With this one. So part 2, what a plant knows. So talking about the fibres, on how it's, I think we went through this stuff. Hemicellulose holds together. What's this actually saying? The slide wire holds the fibres together. I'm not sure what that relates to, but it's hemicellulose, cellulose, lignin, little bit pectin. That's what makes up the cell walls in plant fibres. Maybe saying you can put it back into concrete. Back in the day, they used to put mud and straw and wheat straw, like the dried up grass type stuff. And actually, if you get, the finer it is, if you chop it up, the stronger it is. That's how those mud houses stay around for thousands of years. That's all they've used. Some organelles inside. The vacuole surrounded by membrane. No, okay. Not going to go into that much detail. The vacuole, though, is one of the organelles inside of the plant cell, and that stores things, as you can see here, and that stores all the bad stuff, harmful, because it's got a pH of 5 and cytoplasm 7, so it actually absorbs and stores all the bad stuff. So that's how plants are bioaccumulator. So it can absorb all the things from the atmosphere and all the toxins, like glyphosate, for instance, and it might store it in the vacuole. Then you'll go and break that down. You won't be able to see it inside the plant cells, but it'll still be there in very low trace amounts, and that's what that nano and pico amounts that still are carcinogenic and cause problems. So how's a plant cell elongate? Well, you're going to add water to the cell. It goes a little bit more turgid, so it expands. It expands at each end, and then the cell actually elongates. Turgor pressure provides the plant with much support for structure. And then at permanent wilting point, which it looks like here, that's when the vacuole loses volume and the cell loses its turgor. So here's the cell here. And you can see over here with the cell, it's the water inside is actually holding it up, giving it structure. And when it loses too much water, because the plant's 70% water like we are, or around about 70%, it goes to permanent wilting point, which is stress. So that's when it's stage when it'll start inducing making abscisic acid, one of the phytohormones, and starting to produce drought assistance for itself. So turgor pressure provides the plant with much structural supply. Okay, this must be to show the third slide. After you water it, the cell absorbs all the water and starts to grab pressure again, create turgor, and then it'll stand up. You can see that happen pretty quickly, within half an hour. Cell membranes with 2 adjacent cells are connected. Okay, let's see how they speak by the plasmodesmata. The plasmodesmata creates a continuum of cytoplasm and plasma membrane between two adjacent cells throughout the whole structure of the plant. And it also enables communication and material to be shared between it. Most cells of the plant are connected from the root right to the tip to the root apical meristem. No, from the root, the SAM to the RAM, the shoot apical meristem right down to the root apical meristem. This is the inside of it. Oh, okay, we're up to the vascular tissue. So how the plant gets its minerals and water through the big plants, like the whole massive redwood tree that's 100 meters or 300 feet tall. How does that get water to the top? Well, it's through these tissues. It's a vascular bundle. There's the xylem and the phloem. Actually, we'll go into those details. Vascular tissue. So these are ones in the leaf. C3 through the main vein axis. It's a good pathway. And then through the cells in the side. Stems. This is all different on different species, different genus. That's just a general to show you the cross-sectional area of what they look like. That's why when you cut a stem or a section of the plant, you might see it weep a bit, and that's producing the pressure, root pressure. And that's a good thing. So the ground tissue is the main mass that carries out photosynthesis and it helps support the plant. The epidermal tissue on the outside is the outer covering that provides protection. And you know, while we're talking about the dermal tissue, there's 3 entrances into plant leaves: the stomata, hydothodes, nectathodes. So they're all openings to the air outside where it can all possibly get solution through, like your if you foliar spray, for instance. Here's an electron microscopy image of the dermal tissue with the trichomes, the hairs, like structure. They protect it. Trichomes have different functions. Well, this one has a bulbous head on it. So that would have some sort of secondary metabolite to protect it or to attract it, attract insects. The vascular tissue in the root, okay, I couldn't go into that much detail. Phloem, there's 2, yeah, the phloem of the vascular bundle is 2. The phloem go up and down, and the xylem only goes one way, it's uni. The xylem transports water, and the phloem transports all the sugars. So when people tap into the birch tree to get all the maple tree to get all the maple syrup, that's what they're tapping into, the phloem, to get all that sugars. And they boil it down to make it condensed, like honey-like, because it comes out pretty liquidy and it's not that condensed. So I'm not really going to go into detail of the vascular bundle. Let's have a look, see what people reckon. As Jeff Rowe says, I appreciate that microbes more than ever lately. Topic sounds good. Oh yeah. Hello, Infinite Wonders. How you going? Hello, Lene. Nice to see you. Excellent subject. Yes, what a plant knows is what today is, part 2. But that's microbe 1 with glyphosate. Gee, it's going to be great. Speaking of that professor last night, was just so enlightening. There was no one there even. It was just me and him. It was so good. There'll be more of them. So if you've got any questions, I can ask the professors and they can send me the relevant info to get the most up-to-date. And all those papers that I got too, they're all 2025. Like, I was just so appreciative to have it. It's just been excellent. Xylem develops, no. Xylem develops. Oh, the apoptosis is pretty cool. That's programmed cell death. So when an event happens inside of a plant, it can perform apoptosis and kill a cell off. So that might have the problem trapped inside of the cell, disease or fungal issue, something, and then it can kill it off and just trap it there. So it's apoptosis. Another cool thing for it is a hypersensitive reaction. Wow, that's a big looking spider. Last night shutting the window, it was difficult to shut. I didn't think much of it. Now looking at the window when it's opened, I can see why. There's a big spider about the size of a tennis ball that got crushed in it. Thank goodness I didn't open the blooming screen for it to come in. Anyway, back to this. Hypersensitive reaction is an interesting thing too, to kill off any problems. You might see little spots on leaves and then it starts to grow. That's the little hypersensitive reaction. I'm going to go into the seed elements, and phloem, and the callus. Cytokinesis, no. Disruption making new cells, very good. No. Okay, come on. Some different axes layer. Asymmetry. Meristems are pretty important. That's where it derives all of its new cells from. So there's heaps of different spots in the meristems in the plant throughout its growth cycle. Even when it goes to a secondary growth phase, you get lateral meristems. They're the spots that create new growth cells in the center, in the quiescent center. And then next cells outside that haven't been given a direction yet. This is surely going to go into it. Shoot apical meristem. As you see them up the top and you ram down the bottom, you root apical meristem. It's this one. It's not me, okay. Cell organelles of an animal. Notice it's lacking a cell wall because it's an animal. There's a plant cell with big, thick cell walls, plasma membrane. What's this? Eukaryotic plasma membrane. The eukaryotic plasma membrane is a phospholipid bilayer of proteins and cholesterol embedded in it. Right, I'm trying to make this applicable to the course, so I'm not going to go into detail about confusing you and about what the slides actually explaining what the slides say. This is pretty cool. This is a stain of a cross-sectional area of a stem, I think. Yeah, it's a stem. Oh, cross-sectional area of a stem. Yeah, so you can see there, that's the xylem, the water bits that go from the root to the shoot only, and the phloem, these little tiny cells out here. You can see them all, little tiny ones. That's the ones that go both ways, that link up. Monocots and dicots. The first little leaf that comes out of a seed, the seed germinates, the little radical comes out, and the first leaves, if there's one, it's a monocot, and if there's two, it's a dicot. Is there any point? Going into detail. The monocots are really old school. They're the billions of years old ones, and then the evolution happened and it formed the dicots. So most of the newer plants are the monos, and the old school ferns and bananas and all that sort of stuff are the monocots. They do vary a little bit. You've heard terms called annual, biennial, and perennial, or annual plants. Plants with a life cycle that lasts only a year. Biennial require 2 years to complete the life cycle, and perennial, like fruits, trees, keep growing, put new growth out every year. So if you're going to get into breeding a bit, it's pretty important. Like, for instance, carrots, which I've just mucked around with, they take 2 years to complete their life cycle. So you can harvest it the first year. Yeah, you're going to get your stem, your cortex, which is what we eat, that orange thing. But if you have to wait for a second season to come out to actually get the seeds or make the seeds. Here's the cells in the quiescent center, and how this is inside of the meristem, so apical meristems around here, and these are the cells inside of it. So it first goes and initiates the cells. So it goes and they haven't got any direction yet. And then as it forms, keeps producing more, these get pushed down, and then it starts getting into differentiated, where they get a pathway of what they're going to be. It's pretty cool. So that's for the tissue culture. You can grab that quiescent center and you can make the cells turn into whatever you want, roots or shoots, or just stay into a big chunky callus. That's what it looks like under a microscope. The shoot forming with the pericycle cells of the cells that first form to make the shoots. So it comes out from the top and down on the root. That's the quiescent center in there because there's a big root cap. So if you go in tissue culture, the first bit, you're not going to get much. It's just the root cap where it protects it. In most species, water and mineral uptake by roots. You really want to know about that? There's 3 ways you can get into it. I'm just going to go into it. The epidermis is responsible for selective mineral uptake because of the wax barrier. Minerals cannot go between cells, but only through cells. Epidermis is responsible for selecting material. Okay, let's get into something. Root cap protects the root meristem and reduces the friction by secreting mucilage. There's the root cap there itself. You want to get into the cellular form. So this is the quiescent center. And it comes out from that. Different cells have the different names as they get given tasks. The epidermal cell that borders 2 cortical cells develops root hairs. That's how the rhizophagy system happens, where the root tip goes out, the microbes go out through the root tip and back in through the root hairs at the side of it. That creates a little tiny rhizophagy cycle with the microbes cycling around. And the exudates that the plants put out, little secretion that they do, multi root, mutant root. Just various spots to get the meristem from. So it's really wherever new shoots, new growth is starting to happen, there's got to be some sort of meristem in there to make that happen. Here's the start of a shoot, starting to form little cells, and they've got direction and starting to grow. Chlorophyll. It's the cellular zoomed in bit. It's pretty cool, eh? So the cells, see the dots of nucleus in the middle of them? It's red, that's been stained to get that effect. So at some point, the meristems that have been producing stems, buds, and leaves changes its purpose and starts to produce flowers. So it goes from its vegetative growth to its flowering growth, produces the florigen hormone. Multiple environment and internal cues regulate the switch to flowering. So the day length, the light quality, the plant hormones, temperature, drop in temperature, and different development processes that have happened maybe in its earlier stages, like vernalization or things like that. ABC model, what's that mean? These are just the parts of it, of the carpel, which is the flower. So the stamen is the men, the sepals are the women, and the stigma and the style. Yeah, didn't really go into much detail. What's this? A class of genes suppress expression of C class of genes in hole 1 and 2 and vice versa. Don't know the experiment. A gene that is essential for a formation of a shoot meristem. So when expressed in root cells, initiates formation of shoot apical meristems. This is a tissue culture. So they've gone and put a little bit too much cytokinin in there, which is gone and initiated shoots. It's a cross-sectional area of a leaf. And you need to go into detail there. There's the structure of chloroplasts, which are the green pigments inside the cells there, you can see, which give the plant the overall green tinge. The structure of the chloroplast. Photosynthesis takes place in chloroplasts, which have an outer membrane and an inner membrane. Stacks of these thylakoid lumens called grana, and third membrane layer. That's how they grab energy out of the sun and exchange it and turn it into their adenine triphosphate, which is down here, the ATP. It comes in as ADP, adenine diphosphate, and goes out as ATP. But that's how it does it anyway. The chloroplasts, those little dancing things, if you energize them, you can see them moving pretty good. Not real fast, kind of northern lights, a bit faster than northern light speed. ATP is the molecule that contains a huge amount of energy. It's the currency of the cell, they call it. It's the energy of the cell. Adenine triphosphate. Oh, that's how it's broken down to grab energy out of it for its metabolism, different processes. I'm not going to go into this detail. Electron flow. NADPH is another form of ADP. It's another form of energy. Photosynthesis. Does this happen at bloom night? The answer is yes. There's two stages of photosynthesis. There's a light reaction and a dark reaction. And in the light reaction, it traps the sunlight and stores the chemical energy to be used later. And in the dark reaction, to capture carbon from the air and convert it into chemical compounds, sugars, using the energy produced at the first stage. So that's where through the day it'll be respiring oxygen, and then at night it respires CO2. That switches through rubisco, which is an enzyme that can do two different things. When chlorophyll absorbs light, the energy from the light causes an electron to jump out of the chlorophyll, and the electron travel in pairs. Okay, very good. I'm not going to go into that detail. Question: what is the source of oxygen in the air? What is the source of oxygen in the air? Is it H2O from the soil? Is it oxygen from the stroma or stomata? Or CO2 from the air? So how does the plant get oxygen from the air? It makes up 45% of its nutritional requirements out of the 17 essential elements. So what's the answer? Well, the answer is it gets it from the roots, through the soil, and it gets it out through the water. I should stop actually, see if there's any questions or anything. Oh, questions, been a little bit of chat. Hey, even got a thing open, not wonders. I know of you. Fish and Grow. G'day, Fish and Grow. New Zealand. Nice to see ya. Oh, hang on. Miss Bonnier, Genetic Memory Farm said, I would bet that the average plant knows a lot more than the average human. Just my guess. Yeah, you're probably right, mate. Especially those big redwood trees. Imagine if they could speak. G'day, g'day, g'day. Fish and Grows, how you going? It's a question mark. Oh no. Roots, oxygen from the soil, says GMF. Yes, good. Well said, mate. I thought we were supposed to. Yeah, you were. It was a bit of a, for people who aren't watching the screen, it was, you could have answered that. Like, which one, how do you think that's why, yeah, it was in there. And good work. So why can't I see fish? There it is. There we go. Moderator. So get back to it. You're going for half an hour already. Good flies. So back to it. Here I'm sharing. Good. No, I'm not talking about for photosynthesis or dark reactions. It's pretty cool how it happens at night, isn't it? So that's why you've got to give the plant adequate time to, like you think of outside, and it's pretty rare unless you're up in Alaska or Russia that you're going to get 24-hour sun. So pretty much the plants are used to having their light reaction. They're going to sleep. Sugar beet. No, I'm not going to go into the molecular details. I'm using an experiment, which is photosynthesis. Don't know what DCMU is. Plants were domesticated in several regions. Oh, very good. This might be the, oh yeah. Human intervention has invented all our cultivated foods. You know, all the brassicas come from one mustard plant. Not one, various mustard plants, because to do different breeding techniques, you need a good range. So the cauliflower, broccoli, cabbage, kohlrabi, kale, all come from mustard. Isn't that a spin out? All brassicas, brassica family, Brassicanaceae. Central dogma. Oh, gee whiz, that. No, DNA. Recombination. Actually, I'm not going to talk about DNA stuff. Recombinant, what's this? Again, add cytokinin in for growth media. Gall tissue can persist indefinitely in culture without additional plant hormones. Gall tissue can persist indefinitely. Okay. All right. I think you need to add some sort of other media with that, but there must be more to that slide. Genome transfer is a natural path for formation of new species. Sure is. That's what the whole thing when you're doing for breeding, you want to get new genomes, new DNA, new pathways to make new species. The potential genetic engineering. The bacteria Bacillus thuringiensis, or BT, kills insects due to a toxin that punches holes in the cell membrane. That's anyway, the BT toxin only works in the alkaline environment, like insect guts, so it's safe for humans. Organic farmers spray entire fields with Bacillus thuringiensis, I do too, bacteria to combat insects. So it sits there, and when it's in its favourable conditions with enough moisture, it'll just do its thing, and when the moisture dries up a bit, it'll start and form its endospore at the back of it, shaped like Tic Tacs, and then it goes into spore form and sits there. And then it'll wait for favourable conditions, more so the water to come back in, the temperature, and then it'll germinate and happen again, and just keep repeating. And then if its favourable conditions last for a while, it'll reproduce itself. So that's how the microbial cycle works. So, but if you go and use poison or any type of sides or bad things to kill, bang, cross this off. And more so, you'll be increasing the pathogens. So how can this technology be used for common good? The boll weevil and other insects can completely destroy cotton crops, as an example. So through Bacillus thuringiensis, it fixes it. Oh, that's the last one. Really? I think that is the last one. Let me check. The next one goes into my certificate. Yes, it says my name on it. Fair dinkum. Well, I hope that was a little bit enlightening. GMF says, I'm startled by the amount of growth and movement the plant does in the dark. Can look totally different from lights out to lights on. Let's have a look at a plant in the dark, which it's called etiolated. If the plant has grown with no lights, and that's because the chlorophyll pigment hasn't developed and absorbed chlorophyll yet. I'm trying to look for the website that shows all this time lapse of plants. And we're going to look at that again. Okay. I should just go to last week's. I remember it was in there. I put it down. I'll just have a bit of a quick look here. A lot of bookmarks. All right, 3 more seconds. If it's not there, I'm going to go and look on the last week's one. 3, 2, 1, bang. I love that last week's. Content. Hey, what is there? What do you think about me changing my name and adding at the end of it Aussie CC and organic plant scientist, or organic plant scientist, or what do you think? Something on, because I want to get out of what I was doing, because it's just getting me nowhere and it's just not good, really. It's a shame. I want to get back into agricultural stuff. That's what my training is. So here we go. I got the website, Plants in Motion. So let's have a look at that. Indiana University. Well, I'll get back to sharing the screen. Infinite Wonders says, yeah, YouTube often suppresses content brands. Yeah, it's ridiculous. So what do you reckon? Put an organic plant. What do you think? An organic plant scientist. A scientist is actually anybody who contributes to that industry. So if you contribute to your baking industry, you could be and do new things and do proper scientific experiments in that way. You can be a baking scientist, for instance. Or I can give it a shot, says Infinite Wonders. Yeah, I'd like to put something in, because I do focus on microbiology and soil science a lot too. So I'd like to put something in those, but I don't know how to get that in. But yeah, I need to put something else in, because I'm getting all these shows now monetized. So it's I'm actually getting something out of it, if you know what I mean. Or in the past I haven't been. All right. Because I do all this stuff for free. I put so much effort in. So here we go. Let's have a look at some germination in etiolated conditions under no light. Great, there's none. What about some, you were talking about the way before how plants move and spiral. This is a time lapse over a 24-hour period, and you can see how they're twisting. And the reason why they're twisting, they're trying to grab new growth. So there's no wind in here. They're just doing that themselves. By shifting around the cells in the plants, remember we were talking about them with calcium and stuff before, and oxygen, and you stretch one side and it can make it bend another way. So that's the twisting one. Shooting star, stem. Let's see what this is going to do. See the leaves starting to come up a bit. These ones are going crazy. Looks like they sort of nearly given someone the middle finger. Rude plants. This one might have, see, it's limp like that, so that's probably might have a water problem in there. Well, that could be some sort of issue going on in the genes, heterozygotic circadian responses. Let's see what's under that one. Isn't that interesting? The plant has biological clocks, just like we do. So I think this is going to do with tropism, which is how the leaves go up and they trope towards the light. So it's night and day, comes in. So you picture the leaves as solar panels. See how they're starting to change the direction to the sun, or to where the light source is. There you go, look at this big one, all shut like umbrellas, trying to conserve all their moisture. Bang, let's go and produce photosynthesis and get it all cycling. When nighttime comes back round, oof, we're out of there. Comes back on again, pretty cool. Let's look at some pumpkin leaves and see what happens here. And the night and day cycle through the circadian rhythm. Let's look at the flowers. Watch them open and shut. Oh, already did it. You can see the growth size in this pumpkin. Sure, got some nice watermelons coming on at the moment. That's the next one. General growth. No. Oh, wilting. We were talking about wilting before. So water makes up 95% of the fresh weight of some plants. So see out here, watch the tip, I'll leave my cursor there. See how it's slowly starting to dip down. So it's reached its permanent wilting point already now, and it's starting to close up and trying to, all this stomata is shutting up and trying to conserve all the water that it does still have left inside of it. And that forms that makes the plant go into stress, and abscisic acids form, and the roots start and elongate. So all the growth and development, instead of going into vegetative growth, it goes into putting out more roots, lateral roots and root hairs to try and source water. So it's not a good state for it to be in. That was pretty good. Oh, look, it's been watered. So right now it's been watered, and pretty soon afterwards you'll notice it, and you can nearly see it by eye. So you can see here, leave my cursor there. Oh, that's the end of it. Okay. Root acidification, rosette growth. Let's watch the flower. Oh, that's not rosettes. What's it referring to, rosette? And oh, that's a genus of Arabidopsis. Is it? This goes for a minute. It probably forms a flower. Right, fast forward. Oh, here it comes. Oh yeah, rad. So it zooms right in on the center. And this is a time lapse of 21 days, taken every 30 minutes a photo to make this time lapse. So you can see all the cells starting to change, and then there's this formation. That's pretty cool. Here we go, actually into flowers. Oh my goodness. Sunflowers, this is a big one. They're humongous sunflowers. Oh, wow. Oh, that's wicked. Let's go big screen on this. Oh, we can't. So you can see up here as they're starting to form on the edges, and so the growth is happening from the center here, and it's going outwards, and as it goes outwards, it starts to form in the leaves right on the tips. Oh, that's its reproduction bits. It's not forming a leaf. That's right. What about what else does people know? Lilies. Watch a lily do its thing. Oh wow, look at that. Will it shut up again? How cool. It closes up at night. Isn't that amazing? Moonflower. Don't even know what that is. Does it flower under the moon? Moonflowers open in the evening, so they can be pollinated by night-flying moths. Like most moths pollinate flowers. Moonflowers white, which attracts moths. There it is. Whoa! It's different, isn't it? What else? That'll do for the flowers, eh? All right, click cellular response. What happened then? Cellular response didn't work. Okay. Photomorphogenesis, it's still something here, some power. So photo means light, morphogenesis means visually, genesis means beginning. So what's the difference here? Oh, here, one's in the dark. So this would be growing in etiolated. Let's say the word down here. It lacks chlorophyll. It's EIOLATED or something like that it is. And then as soon as you put it in the sunlight, it'll start to go green. It's pretty cool how plants work. Let's watch that again. So yeah, this is germinated in the dark, germinated under the light. So absolutely no difference. If anything, the left one will stretch more because it's looking for more light to produce more energy so it can start. You see how it hasn't even really opened yet? These have opened the cotyledons, which is the first energy storage to start. That's it. We watch tropisms. That's how they track the sun. Shoot. Well, actually, corn would be pretty good. If it's a tall plant, it shows its tallness. Oh, shows the tip of the corn, really. See it growing? Oh, that's the root. See how it's gravitropic? Oh, that's what it says. Which means that this is laid on the side and they tested to see if this root is, does gravity have an effect on roots? Well, yes. So they put it on the side and see if it starts to go down, proving that it does. All right, I think that's about it, isn't it? That's about it for What a Plant Knows. It was a fascinating course. If you'd like to do it, put it in your search engine. It's a free course. Well, it was when I did it. And yeah, it's pretty interesting. It probably goes for about 6 hours odd. There you go. That's the summary of What a Plant Knows. So I'll put that in the description again below. And wrap it up for that. Dig the time lapses, says GMF. Yeah, they're great for understanding, aren't they? Then we can picture what goes on at different growth stages and we can know. So we think, oh, geez, if this is going to happen, oh, we better do this, or we can do that. Or, yeah, I like the time lapses. Plants in Motion from Indiana University. And next week, how about I just do the glyphosate thing next week? Here, Infinite Wonder says, great stuff, very interesting, thank you. Refreshing to hear from someone that actually knows something about plants instead of growth science. Yeah, thanks for the nice words, mate. I still study at university, so I'm getting a better understanding as the years go on. So it's very rewarding for me to go through my slides because it reintegrates my understanding of it. And because I still do ongoing research in plant science, I'm getting into microbes highly now and starting to get them to work for me, do different tasks. And it's just fascinating because most of this stuff that I learned at university is really, it works. But like putting all the resistance into the genetics, and now this crop breeding course, actually try hit the thumbs up, people that will help. Good on you, GMF. Yeah, thank you, mate. It will help me a lot. So I don't get shared enough. Here, I'll show you. Get a load of this course I'm doing. This is, I'll eventually talk about it. It's crop breeding. It's called the basics in crop breeding. It is so not basic. Holy mackerel. I'm doing pretty good in it. I hope to pass the exam. No, that's not it. this one. So check out some of these slides. And there's, actually, where's a way for me to summarize it? There's so many different ways for breeding. And this goes into what type of outcome you need to what type of process you want to do. Because when you mix things at different stages of their genetics, you get different outcomes. Like talking about homozygosity and heterozygosity. Then you've got your hybrids, different ratio. I'll just skim you through a few just to show different outcomes from different experiments when doing at different stages. And these are all morphogenic expressions too. So they're visually, so you might do round or wrinkled, and it might be a color thing. So there's a few different ways to do these experiments. You do test crosses at different stages. Then you want to grab a particular gene out of a plant. You can. You just keep crossing it until you get that morphogenic expression, back crossing it. That's this extreme easy summary. Or you can do in pure line theory if you want to try and then the latest is to use a chemical to make it homozygotic in 2 seasons. It's really, really, really interesting. I'm finding it. Talks about masculation, how you have to remove the male bits to pollinate to the female bits, and why you want to do that, different stages. This is look at this lovely sheet for a test cross. So what do you want at the end? So you've got tall dwarf. How do we make it all dwarf? How do we make it all tall? Well, you can. How do you make it one color? Well, you can. How can you make seeds all big? You generally can. Pure line. There's different ways. Pure line is one technique of growing plants to purify the line. Is it long story away? Actually, I just find this really interesting. That's another theory. Let's go back. I shouldn't. Back cross breeding. Another way. Why do you want to back cross? Well, that's when you want to really grab the gene of interest. There's different ways to do it for the homozygosity or the heterozygosity. It's the stability of the genetics. So yeah, conventionally you do it in 6 to 7 generations. Back cross for a dominant gene transfer. But you can do it in 2 generations in late days. Or here, if you want to do a recessive gene transfer, so you might want to have that in the background. Anyway, I'm thinking of, what's this one? Merits and demerits for back cross. I love this stuff. Oh, and then you wouldn't believe what is in the next week. It's all about polyploids. Here's some questions. This gets a little bit specific combining ability. So you might get the combination, when they go through recombination, are they actually combining? Types of recurrent selection, different selection techniques to pull out the phenotype of interest. And he explains too how to do it. So I've got such an excellent understanding of why you do it and at what stage. I love study when it's organised and from an excellent professor. He is so knowledgeable, this fella. I have to slow it down sometimes. He goes too blooming quick. Yeah, the ploidy. Where's the ploidy stuff? Oh, heterosis. That's hybrid vigor. And inbred depression. You've all heard of inbreeding. Yes, you do get inbreeding depression. That's what it's called in plants. When you keep inbreeding them too much. Anyway, I'm not going to. But the fascinating bit was all of the polyploids, doing stuff about the alloploids and autoploids. It was, haven't been, I've got quite a lot of slides on them because I've been doing the research for quite a few years. But just to get the latest stuff, this course, that crop breeding course was written last year. Oh, here we go. Just thumbs up. Good on you. Cheers. Thanks, Aussie Jeffro. Appreciate it, mate. Genetic Memory Farm says, love my microbe army. They have a worm and isopod transport network. That's the way. He's into his organics too, this fella. He knows his stuff. Mendel's peas, yes. Gregor Mendel. They were his pea experiments that he did back in the 1800s. That was the first slides that go into all of the history of breeding, and it goes heavily. It goes nearly year by year from the 1600s on breeding, on crop breeding, plant breeding, and how it's evolved. It's just blooming excellent because we can all use that to our advantage. For instance, if you're into your health, well, you can make your own seeds for your fruit and veg that you grow, specifically your vegetables, because fruit takes many generations. But, well, what did I do last season? I made seed for broccoli, cauliflower, Brussels sprouts, carrots, and what's that bean? Kidney beans. So I've got all of those. So what they'll do, they'll grab the epigenetics. So all the stresses that they've been, I'd put them through. So the different soil structure and the elements that are in the soil, my organic practices, how it virtually gets starved through its different growth stages because it's not getting given what it wants. It has to search for it and ask for it because it's etchugates. So that's the epigenetics coding into that genetics. So then I'll plant the seeds I've already planted the next season, and then they'll grow up, and I might do it again, or I might wait for another season and then make seed. So I've got a lot of seed of the brassicas, that's for sure. Like 2 carrot plants produced, I'd say 20,000 seed. 2, and when you can smell the secondary metabolites for the carrots, and it smells very sweet. So I know that this is going to be a good offspring. So a couple of good expressions that you can pull out through it that can really benefit us as humans. So by these courses and this information, it's not only gains us in our love or hobby that we have, you can also produce highly quality organic food for you and your family and your neighbours, and keep on going when you produce it through the sustainable practices that I like to portray, talk about. I don't like the mineral feeding, but anyway, I won't go on too much. You can tell that I like the breeding and the microbes. Just love it. Love it. It's fascinating. Only 1% of microbes has been discovered, and it's pretty only in the last 100 or so years, and since the microscopes come out. So it's a relatively new field, if you can look at it like that. It's rad. And then you've got the combination of them. Oh, anyway, I better. So next week, what do you think? Should we talk about the glyphosate with microbes? And then the week after, something else. But because it's going to be fresh, I'm going to be reading all of the papers through the week and have a lot of information on it. And even the little bits that I've read already is interesting to the human, because even if you're in the United States, you might want to stay clear from certain brands of certain wheats that have a higher amount of glyphosate in them, for instance, or Roundup, as commonly known. It's toxic, goes through systems, and it's horrible. So what do you think? We'll do that next week? All right, I can reword next week's one to glyphosate on plant health, human health, microbe health, and soil health. It's not looking good. I'll write something like that. So hopefully some interested folk will tune in and we'll get some good discussion on the topic, because it's fascinating to search that in Australia and find very little, or find very favourable things to say, no, there's not much on it. No, it's pretty healthy. It's, yeah, we should use it. Yeah, because the government pumps it out. Yeah. Aussie Jeffro reckons sounds good. All right, good on you, Aussie Jeffro. Thanks for the feedback. And then if you cool viewers could think of something for the week after, I'll be open to topics again. Maybe we can go through another course, or maybe we can do a few. I know there's been a couple of questions that have been suggested. I think Aussie Jeffro and Genetic Memory Farms both have suggested ones. So maybe we could touch on both of their topics that they wanted. That might be the show for the following one. Yeah, I'm trying to keep it active and interactive. Far out. Righto, that'll do. Thanks everybody for listening. I hope to see you next week, and we can discuss to look at our health and how we're being blinded through society from possible restriction of search engine use of different things. Happy breeding, happy growing, good health to you all. Thank you for listening, heaps. Bye-bye. Oh. Well said, mate. Bye-bye. Heaps. Thanks again for contributing.